Hi, I'm Derek Jensen. This is Resistance Radio on the Progressive Radio Network. My guest today is Drew Harvell. Drew Harvell is a professor of ecology and evolutionary biology at Cornell University and resident at Friday Harvell Labs in Washington San Juan Islands. She's author of over 170 scientific publications and author of the award-winning A Sea of Glass and just released Ocean Outbreak. Her research has taken her from the tropical waters of Hawaii to Mexico to Indonesia, Australia, and East Africa. Her current work is on ocean health in temperate waters of the Salish Sea. So first off, thank you for your work in the world, and second, thank you for being in the program.
Well, hi, Derek. Thanks a lot. I'm delighted to be joining you here.
Oh, thanks. So who are sea stars? And let's start with them. Who are sea stars, and what has been happening to them the past decade?
Well, my background is as an invertebrate biologist and a marine ecologist. And so I study a lot of the spineless critters in the ocean that kind of drive the big ecological interactions and make things happen. And sea stars actually are a really important component of our ocean ecosystems. Some of your listeners might usually call them starfish, but they're not actually fish. So scientists have been more recently calling them sea stars. And that's just that whole group of beautiful critters that are star-shaped in the ocean. And the reason they get a lot of attention is because some of them are what we've called keystone predators. And that was a term that was coined, maybe 50 years ago by a former mentor of mine, Bob Payne, to describe the ecological importance and the fact that sea stars in fairly low numbers can control what the seascape looks like, just because they're voracious predators. They're kind of like the lions of the deep, and they eat a lot of their prey and can control their numbers. A main focus of my new book, Ocean Outbreak, is about one of the largest disease outbreaks we've had in the ocean, which affects sea stars on the West Coast and expanded to some of our other coasts.
Back up just a little bit, and whom do sea stars eat primarily? So whom do they eat, and how does that? One reason the buffalo are called a keystone species is because their wallows end up becoming habitat for everybody else. The same with prairie dogs. So can you tell me just a little bit more about their status as keystone, or not their status, but their role as keystone species?
Oh yeah, that's a great question. Well, in the waters that I'm working in now, which we call the Salish Sea, which includes Puget Sound and the waters north of there to the top of Vancouver Island, so into British Columbia, there are about 85 different species of sea stars on the books that might be in our waters. Sometimes some of them are rare, some of them might be gone, but there's a lot of different ones. And they all eat different things. So our very common intertidal star, the one that, anybody on the West Coast would have seen, is called the ochre star. And that's actually the one that was originally called the keystone species. And that one eats lots and lots of mussels or clams if they can get them, but sometimes they'll eat snails or chitons. So, but they're capable of controlling the whole level of the mussel bed. One of the other stars that we've worked on is called the sunflower star, and that lives subtidally, and it's the largest star on the planet. It's the size of a manhole cover and has 24 arms, and that one eats lots of clams, but also sea urchins. And so in places where it's abundant, you don't find as many sea urchins because they can control their populations. And similarly, when you take that star away, sea urchins can become much more populous. Some of the other stars are stars that eat sponges, all kinds of different critters in the ocean.
And when you take the urchin thing, because I think about that urchins, if I'm correct, eat, uh, what I've heard about them is that they eat a lot of kelp. And so without the sea stars, there can be an overpopulation of sea urchins, which can lead to a degradation of kelp forests. Do I have that correct?
Yeah, Derek, you do. And that's actually the subject of a lot of interest and concern right now because the sea star wasting disease epidemic that started in 2013, so that's 6 years ago, pretty much wiped out populations of the sunflower star from Mexico to Alaska. And in the last couple of years, in particular locations, particularly in Central California and Northern California, there have been massive outbreaks of sea urchins caused by the decline of the sunflower star. And so it's an example of, an ecological interaction really controlling the distribution of a major habitat, the kelp beds. And those kelp beds, of course, are important because they're habitat for a lot of our nearshore fisheries, abalone, crabs, and other organisms. And so the demise or the denuding of those kelp beds by the urchins is causing economic impacts and is of great concern.
That makes me think a lot of the role of wolves. And, since they reintroduced wolves into Yellowstone, that has improved trout habitat and insect habitat because without wolves, the grazers, elk, deer, browsers, got really lazy and would hang around the riverbeds. And that meant that there was not the next generation of willows, etc., were not able to get started, which meant the degradation of I can't remember the word, the habitat next to a stream and riparian zones. And so then the wolves came back in that chased the deer and the elk out of the best habitat, and the trees were able to recover, which meant beavers could come back in and they could improve the habitat for the fish. So it's kind of weird. You wouldn't think there's a relationship between wolves and fish, but there is. And it sounds the same, that it sounds, like you said earlier, they act like lions. It sounds like wolves too.
Yeah. Yeah. I mean, I think it's very common on both land and under the sea for major predators to have an enormous impact on all of what we call the trophic levels below them. And so this is there's a word for it. It's called a trophic cascade. Ecologists like to have terms. And that trophic cascade can be run by either a sea star or a wolf, or in some cases, sea otters too. So there's a lot more general examples of that.
So you mentioned sea star wasting disease. Can you talk about that, please?
Yeah. So as a marine ecologist who specializes in disease, some people call me a disease ecologist, and specifically infectious disease, I've been running a network called a research coordination network of disease ecologists to study the different outbreaks in the ocean. And so we've been focused on critters from oysters to seagrass to corals to dolphins to sea stars and turtles. So, there are lots of organisms in the oceans where there have been big outbreaks. But, the sea star wasting disease outbreak that began in 2013 was a very unusual one in that it started quite suddenly and unexpectedly. And it ended up being bigger than any of us had seen before. And by bigger, I mean that it affected more different species of stars. So, for example, there were over 20 species that were dying in what we call a multi-host outbreak. And the geographic range was unusual in that it spread all the way from Mexico up into northern Alaska. The outbreak may have begun 2 years earlier on the East Coast, and eventually would extend further into the Pacific, into waters of Asia. So it's been called the largest disease outbreak of marine or ocean-related wildlife because of that. And so what we initially saw were reports of stars dying on the Olympic Peninsula and north of Vancouver and Howe Sound. And the initial reports were from divers who saw vast numbers of the sunflower star dying in kind of extreme grisly ways. So their arms were falling off, their organs were spilling out of their body, and they were dying in really high numbers. And that began in June and August of 2013. By October of 2013, the epidemic had spread and was again unusual in that all three of the major aquariums on the West Coast, so that's the Vancouver Aquarium, the Seattle Aquarium, and the Monterey Bay Aquarium, reported within 2 weeks of each other massive mortality of all of their captive sea stars. And so that got everybody's attention because many of those collections were over 20 years old. These stars are pretty long-lived, and so that had never happened before. And so that kind of added to the concern about what was going on with this outbreak. So we started working on it in December in Seattle when there was a massive spread of mortality and dead stars showing up on people's beaches. And we began doing our surveys then.
Oh, actually, I have another. I want to talk a lot more about the disease, but before we do that, I just want to clarify a little bit. So when we have 20 different species of sea stars, if we made so, would that be the equivalent on land of? Would they be related to each other? Would that be like 20 different species of primates getting the same disease, or 20 different species of, how closely, sorry if this is a really ignorant question, but how closely are sea stars related to each other? Compare them to humans. Is it like humans and gorillas, humans and chimpanzees, humans and lemurs? How close? I'm sorry again for the terribly ignorant question.
Well, it's a little bit of a hard question because I'm an invertebrate biologist, so I'm not really that expert on the relationships among human and primate groups. And so I could really be making a mistake here by saying what I'm saying, but I'll give this a try. There are lots of different classes of different groups of sea stars that the different species fall into. So there's a lot of variation that's covered by those 20 species that are affected. So they weren't just the 20 that were most closely related. And I suppose it certainly might be, let's say, so that I'm a little bit safe on this, it certainly could be like 20 different species of gorilla, right? There's a lot of different kinds of gorillas. And so it's as if most of your gorilla species all became sick from the same thing. And, we know that there are other diseases that are called what we call multi-host pathogens or diseases. And perhaps one of the really good examples for anybody that knows about frogs is there's an outbreak of a fungal disease called the chytrid fungus that's causing massive extinctions of frogs worldwide. And they're estimated to be perhaps over 200 species of frogs that have been decimated and maybe driven to extinction. Sometimes it's hard to know when something's gone, if it's really gone, but definitely decimated. Or, for example, another example with birds is avian malaria caused extinctions of multiple bird species in the Hawaiian Islands. And so, again, to have a disease that affects multiple species is not uncommon. And then just to kind of go on and on a little bit about this, there are also diseases that humans share called with wildlife called zoonoses. And so, rabies, for example, would be one of those. Does that clarify a little bit more what you were? Yeah.
What is the current status of the sea star wasting disease? Has it run its course and they're recovering, or is it continuing to decimate, or what's the status?
No, it's continuing. So we are now 6 years into this. It's 2019, and this started in 2013. In the year following the start of the outbreak, we lost between 70 and 90% of the animals in the intertidal. So those are the ones that we can reach at low tide and count. Of the ones in the subtidal, those are the ones that can only be reached by divers or dredging or some kind of sampling. That massive sunflower star has been decimated well over 90 to 95%, almost throughout its range. And unfortunately, the mortality continues. So I receive reports from divers and samplers pretty regularly. And 2 weeks ago, there was 50% of that ochre star was reported to be affected by the wasting disease on Whidbey Island, which is near Seattle. We did surveys on the intertidal stars a couple of weeks ago, and they actually looked quite good. So we only saw one diseased individual out of, I don't know, I guess maybe 200 or 300 that we had counted. So that was good, but the numbers are still low. But it's kind of a tale of two stars is the way I like to talk about it, because some of our stars are looking better, the intertidal one, but the subtidal giant sunflower star is not recovering, and we're calling it imperiled across most of its range. And that star used to be as common as a robin. I mean, you would see it on every dive practically in our waters. And right now, the only place we're really seeing healthy populations are up into Alaska, which is the northern part of its range. It's what we call an endemic species because it only lives on our coastline. And so if it doesn't return, then it will be sort of not present on our planet. So we're very concerned, and we've actually called a few meetings to discuss whether there are management actions that would be important to aid recovery of that species. But of course, as long as the disease agent continues in the water, there's not really a lot we can do to aid recovery unless we can find ways to bolster their resistance to disease.
Before we talk about to help the populations recover, what do they believe, or what is believed to be the agent of this illness?
Derek, that's a controversial topic right now. There are a lot of knowledge gaps surrounding the causative agent of this outbreak, partly because it's been very difficult to work on, partly because it's novel, partly because it seems to be something small like a virus that's poorly studied in the ocean. So we did experiments in 2014. This was work done by a postdoc in my lab and one of my grad students to test what we would call the efficacy of a viral-sized fraction on stars. And those were experiments to actually inject healthy stars with a filtrate from the sick ones that was only very small, smaller than 0.2 microns, which is typically virus-sized. And what we found in our experiments is that virus-sized fraction made stars sick and die. And then the controls for that are to heat kill that fraction so that it's got all its same components, but it's no longer living. So those were really good, well-replicated experiments that provided strong evidence for the role of something in the virus size range. And to date, we don't really have evidence that that's not what it is. But we really wish that we could have run those experiments on more of the other species and continued to do work on it. But there was very little funding for this kind of research. And, it's actually very difficult to do those experiments once all of the stars in all of our waters were infected. So it only kind of works well to do those experiments with clean populations. So in summary, just to say, our best evidence is that there's a virus-sized microorganism causing the disease.
You said that there are still healthy populations in Alaska of that, I believe it's called sunflower species. And what about areas? Are you seeing any areas where there seems to be some sort of recovery or some sort of resistance developing to this disease?
Well, we think the fact, even though there aren't huge rebounds in our intertidal populations of the ochre star, the fact that there are stable numbers on the shore and that we're seeing quite a few of them does suggest that they must have some level of resistance, or that at least the survivors do. So I think there's, reasonable evidence for some resistance in some of these species. The sunflower star, though, that's not the case. We haven't seen large numbers of them recovering or remaining on the shore, at least in our southern waters in the continental U.S. Now, up in Alaska, water's a lot colder, and so, for example, north of Ketchimek Bay, the waters are colder than 9 degrees, and since temperature is also a factor in the susceptibility of these stars to the disease, we don't really know if it's just that the water's so cold the disease isn't effective, or if it's actually just not there. But those stars are doing pretty well.
One more sea star question before we start talking about warming water. What's the typical life cycle? I'm really wondering, are the purple ochres that I'm sorry, I'm so bad with nomenclature, but the intertidal species you're seeing that are recovering some, what is their life cycle? Are these ones who have survived the initial onslaught, or are these new babies who have come on? How fast do they grow from? And don't sea stars do a thing? How do they reproduce? Don't they throw the sperm and throw the eggs into the water and hope they meet?
Yeah, that's kind of what they do. Great question, because it's an interesting one. So there are male and female stars, and the male stars make sperm and the female stars make eggs. And as you say, they do what we call free spawn, which means they release their reproductive products into the water. And so somewhere in the water column, those meet, they make an embryo, and that turns into a little larva, which then has to feed for 4 to 6 weeks in the plankton and grow up. And after that period where it's been feeding and gotten bigger and bigger, then it's ready to come back to the shore. And so they come back and they metamorphose into little, what we call juvenile starfish, that are actually really quite tiny. They're only about, I would say maybe a half an inch or less when they settle onto the shoreline. And then those grow up, and maybe between, 4 to 6 years later, they can become reproductive and then start to, make more babies. So that said, that means that the ones we're seeing on the shore that are, 6 inches to 10 inches in diameter must be survivors of the epidemic, because they're certainly older than 5 or 6 years old. And then the little tiny ones, of course, would be the new recruits. So that's how we know that some of the stars we're seeing, those intertidal ochre stars, must have survived the outbreak.
And are you seeing, for at least that species and some others, are you seeing a fair amount of recruitment, or are the babies not coming back?
Well, those patterns have been a little bit confusing because following the big outbreak, so in 2015 and even into 2016, there was huge recruitment, like nobody had ever seen before. So many of those little recruits hit the shore and started to grow up, but then they didn't make it. A lot of them didn't survive. And so there was a lot of hope when we first saw that recruitment that everything was going to be fine and this was just going to all be over. But unfortunately, those juveniles also succumbed to the outbreak. So not all of the new recruits are surviving, but some of them are.
Again, before we move to the larger effects of warming waters, what would be, I know that the crystal ball is always cloudy, and especially when speaking to a scientist, the crystal ball is especially cloudy because of, we prefer, I'm a degree in physics too, preferring to deal in sort of facts rather than projections. But what would be the most likely, I guess two questions. Without intervention, how do you see this moving forward? Probably, sort of most likely scenarios. And second, what interventions are possible in the oceans? Again, leaving off stopping global warming for now. We'll talk about global warming in a moment.
Yeah, stopping global warming would indeed help quite a lot. You're right, and we'll get to that. We don't have a lot of experience with managing disease in the ocean. On land, the typical management actions are to cull, that is, you remove all the diseased individuals from a population, and so they don't transmit as much disease, or quarantine, so you keep the sick ones separate from the healthy ones, or vaccinate, you develop vaccines that can be used to fight against future disease. None of those are options in this case, or for most of our ocean critters. And so in my book, Ocean Outbreak, I do talk about how we have a bigger problem in the oceans. One, we don't see these outbreaks coming. Often we don't detect them as soon because they're under the water, so they're out of sight and out of mind. And then secondly, we have really very few options for actually managing across the expanse of our oceans when these kinds of outbreaks start. So we really don't have a lot of options for actively managing, especially in a case with an outbreak like this that's a novel organism that we don't even really know for sure what it is. So our hands are kind of tied on that. That said, we feel that the prospects are reasonably good that these intertidal stars are going to come back, that there's evidence that some of them are resistant, and the natural course of things. I mean, really, Derek, right, this is an age-old battle between hosts and their diseases or parasites, and this has been going on for millennia. So one of the natural courses is that the host will develop resistance, and we hope for that. In the case of the sunflower star, where there is no evidence of that happening, there is the possibility that the current endangerment could lead to extinction. We don't think that's going to happen, but we can't rule that out. We are considering what kinds of recovery efforts could be helpful. So one of those is captive breeding. It turns out that it's not really that difficult to breed sea stars. It's just a matter of fertilizing those sperm and eggs and growing up the babies. And so there are efforts underway, actually right now, funded by the Nature Conservancy at Friday Harbor Laboratories, to begin the process of developing captive breeding for that sunflower star, so that when the pathogen maybe becomes less virulent or less damaging, or the host becomes resistant, we would actually have some captive populations to begin to restore or help along the natural process. The other thing that I think is really important is investigating more the natural mechanisms of resistance. Oh my gosh, we do not know very much about how sea stars resist disease. You can imagine. And so that turns out to be an important knowledge gap. We need more information to begin to understand how natural resistance develops, so that maybe we can help move it along with some kind of assisted evolution or assisted breeding. And then finally, another big priority is more work on that causative agent, the actual microorganism that's responsible. Once we know exactly what virus that is, there's a lot more potential for better surveillance.
So what is, in your perspective, what is the role of warming waters in the spread of this particular disease? And then after that, I would like for you to open it up to your larger exploration of what warming oceans mean for ocean life in terms of the spread of infectious diseases, not just sea stars, but across other species.
Well, in the case of the sea star epidemic, the largest declines were associated with warmer waters, and we actually did lab experiments in the summer of 2014, so near the beginning of the outbreak, to grow that ochre star at 4 different temperatures in the lab. And what we found is that at the warmer temperatures, they died faster. And so while there was no real evidence that the initial outbreak was triggered by a warming event, there's a lot of evidence that the warming events that followed made the damage or the mortality from the outbreak a lot worse than it would have been. So what I like to say is the warming really fueled probably a larger outbreak than we would have seen. And we recently published a paper also about the level of mortality in that sunflower star. And even in the deeper waters, when there was anomalous warming, there were higher levels of mortality. So that's kind of our evidence that warming was associated with the magnitude of this outbreak. And of course, those years of 2015 and 16, our waters experienced what we call a major heat wave. So anomalous warming between 2 and 6 degrees warmer than normal. And that was all the way up into Alaska.
So do we think that the warmer waters make the disease agent happier and more populous, or do we think that the warmer waters stress the sea stars and make them more accessible to opportunistic disease agents?
Yeah, thank you for the very careful way you worded that, Derek. Again, it's one of those areas where we have a pretty big knowledge gap. We can't say whether the warming benefited the pathogen and made it what we would say more virulent or grew faster, or whether the warming partially stressed the host and made it more susceptible. So that's typically what we see in a host pathogen interaction with warming, is there's kind of a double whammy, that it can both make the host more susceptible or stressed, and that it can give the microorganism an edge, because of course a lot of these microorganisms have such rapid turnover and rapid generation times, and a little bit of warming can fuel much faster growth. And then there are even some viruses that are triggered by warming. They're totally not. They don't have any action when it's under colder temperatures, and above a particular temperature, their activity gets triggered. And in this case, we don't really know. All we know is that things are much worse with the survival of the stars under warmer conditions.
So one more quick thing before we switch to the larger global warming question is this also affecting sea stars who lived in waters that were already warm?
No. Some of your listeners may know about the crown of thorns star, which lives in Australia and is a major pest because it eats coral, and it can have its own population explosions that can decimate vast areas of coral reefs. So wouldn't it be nice if that star had been affected? Oh no, that was the one that was totally resistant, and for some reason the tropical stars were not affected by this. So it was only sort of in the colder temperate waters, which I know sounds a little bit I don't know if ironic is the right word, but it is strange that here you have this sort of heat-fueled epidemic, but it's affecting temperate species and not tropical ones.
How does this, we have about 13, 14 minutes left. Can you talk about, can you talk about your book a little bit, Ocean Outbreak, and also the larger question of of diseases, of the relationship between global warming and disease outbreaks in marine environments?
Yeah. Thanks, Derek. Yeah, I'd like to talk about that. A lot of our work we're ecologists, so a lot of the work in my lab has been with what we call either ecologically important species. In some cases, they're foundation species like corals and coral reefs, or the work we're doing in the Pacific Northwest is with seagrasses, or with sea stars, which are keystone species. So we're always concerned about these disease outbreaks that can really have ecological consequences and affect potentially the balance of our marine ecosystems and even the sustainability of those ecosystems. In addition, virtually all of the outbreaks we've worked on are things that are fueled by warming temperatures. And so in the book Ocean Outbreak, I focus on only 4 major outbreaks. And the reason I did that is there are so many. Almost every organism in the ocean you can think of has at some time or another had some kind of an outbreak, and some of them have been very important. And so that was just too big a story to be able to talk about. So instead I decided to focus on 4 that were really important, or that gave us an unusual power to tell the story about the role of disease in marine ecosystems. And so the 4 I chose are corals, sea stars, abalone, and salmon. And I chose each of those for a different reason. The corals we've worked on for many years, and in a way they're kind of the poster child for big impacts from disease of warming, because many of your listeners probably are familiar with the big impacts called bleaching on coral reefs, caused by warming oceans that disrupt the symbiosis between corals and their symbiotic algae. But the thing is, after that bleaching event, the corals are susceptible to further bouts of infectious disease. And so that's kind of what we've been studying. And unfortunately, they've been lethal, causing massive decline of coral cover around the world. In the case of sea stars, again, the point was to tell the story about a sudden, unexpected outbreak that could affect so many species, and they could actually have ecological cascading effects. Now, the case of the abalone, that may be a little bit of a less familiar critter to some of your readers, but those are a one-shelled shellfish that are really good to eat, and there's quite a high diversity of them in the California coast, and they used to be an important fishery. They've been driven they've been driven to endangerment by a combination of overharvesting and a disease. And the disease story is kind of interesting because it took almost 15 years to figure out what was the infectious agent causing massive mortality of over 4 different species of these abalone. And this is another case where these marine organisms are now driven onto the endangered species list, partly by the effects of this disease agent that can affect multiple species. And I should say, just to back up about the corals, several of our Caribbean coral species are now on the endangered species list, also driven there by infectious disease. And then finally, I wanted to talk about salmon because they're important food for us humans. And also there have been large global outbreaks, which we call pandemics, of an infectious virus that's taken out a lot of our cultivated salmon, which are the Atlantic salmon that are grown in big fish farms. And so that is an outbreak that had huge economic implications and really even threatened the ability of farming these salmon until, very interestingly, scientists figured out ways to control those outbreaks and to start to develop vaccines and resistant salmon and better hygiene practices for growing up the salmon. So those were the main ones in that book. And then really the whole reason for writing this book was to not just catalog everything dying and talk about these grim stories, which really are pretty depressing, but let's start thinking about how we're going to clean things up and do a better job with the health of our oceans. And we've recently been doing work in Indonesia on very sewage polluted waters near coral reefs, but that also have seagrasses. And what we showed in our study several years ago is that seagrasses can reduce the levels of pathogenic bacteria by 50%, at least in those waters. And so it kind of gave us the idea that, wow, there's some natural marine habitats that have important hygiene or pathogen reducing services that are not only important for things like corals and sea stars, but probably humans too, because some of these pathogenic bacteria are also dangerous for us. And so there are very large knowledge gaps about how disease processes and transmission occur in the ocean. And there's a lot of room for innovation in understanding natural controls on some of these pathogens in the ocean. And so that's some of the work that we're doing now in the waters of Puget Sound, also to look at the ecosystem services for filtration and hygiene in this case, it would be our eelgrasses.
So maybe this question, but what is the actual mechanism? I know this happens with marshes too, that there are places that cities, Arcata, I live in Crescent City, California, on the coast of California. And south of here in Arcata, they use a big marsh to integrate a marsh into their sewage treatment plant, and it evidently cleans a lot of the infectious agents out. How would that actually work? So you have some infectious agent that is carried along in some poop, and then how does the seagrass actually clean that out? What happens?
Great question. And as you point out, Derek, this is not news that coastal vegetation can have important filtration services and clean our waters. What was new in our study is actually to identify that pathogens were, which kinds of pathogens were actually removed from the water and how many of them, and by kind of a natural ecosystem. And we don't yet know the mechanisms. We think there are 4 that are probably all operating, and it's going to be exciting to pursue the question of their relative importance. But for one thing, in a lot of these seagrass beds, there are natural filter feeders. So there's a lot of clams and oysters and sponges and ascidians and sea squirts that naturally will be filtering some of these bacteria out of the water. And I should say we only did study bacteria. So that's one level of defense. Secondly, the seagrass itself probably acts as a passive filter and captures or removes a lot of bacteria. Third, the way that normal, our septic systems work is by using oxygenation, and a lot of certainly the bacteria that are in human sewage are what we call anaerobic bacteria. They don't like oxygen. Oxygen is toxic. And so just the production of oxygen by these seagrass beds or coastal vegetation can be very important for controlling levels of bacteria. And then finally, one of the sort of more innovative, huge knowledge gaps in this is the potential role of the actual microbiome of the seagrass itself. Now, probably a lot of your listeners have heard about how important the human microbiome is. That is the kind of bacteria that live in our gut. But all organisms have associated bacteria, some of which are very good. And so the bacteria or the microbiome that live on the surface of those seagrasses can also be playing an important role in sort of doing in some of the bad ones. So anyway, in summary, there are 4 potential mechanisms, and we don't really know yet the relative importance of them.
So we have maybe 3 or 4 minutes left, and before we do a wind down, I just have to say that even though you are very, very calm and very clear, that this interview is scaring me almost as much, perhaps, as any interview I've ever done, because the oceans are so big and they are changing, and as you said, we can't see a lot of them, so there are a lot of changes that are perhaps happening that we don't have direct observation on. And so I'm just finding this notion of a warming ocean and pathogens, some pathogens perhaps being encouraged by a warming ocean. I'm finding that very scary.
I think that's fair enough. I think it's a potentially explosive problem, and that's again why I wrote this book, is to, it's a book written for a broad audience. I hope some of your listeners will read it. And I think it is, I don't want to panic people, but I do think it's a potentially scary situation. It worries me a lot, because I care deeply about the sustainability of our ocean ecosystems. And diseases, frankly, scare me because they have such, explosive power and potential. And then when you start dealing with an ecosystem like the ocean, where there aren't really very many boundaries or borders, and it's impossible to control something once it gets in the ocean, I do think that we need to put a lot more resources into working on this problem of both studying how our inputs from land can be increasing diseases, because as we've already said, once something gets in the ocean, you're kind of in a tough spot. There's not a lot to do. So I think that really raises the ante on being much more careful about what we put in the ocean, how much more careful we need to be about aquaculture. We have a major concern about feeding the future. We have a lot of concern about having enough protein, and so that's driving more aquaculture in the ocean, which I'm not saying is a bad thing. What I am saying is vital is that under those conditions, we have to have absolutely stringent best practices to be careful not to have spillover and infect our natural biota. So yeah, I think, and then we're not going to be changing the warming anytime soon, even if, which I hope we'll be dialing back the level of carbon dioxide emissions, but we still have a lot of warming in the pipeline. So we're going to be dealing with an increased level of disease outbreaks for quite some time. And so I think we need to up our game to be able to handle it better.
Well, thank you so much for your incredibly important work in the world. And thank you for being on the program. And I would like to thank listeners for listening. My guest today has been Drew Harvell. This is Derek Jensen for Resistance Radio on the Progressive Radio Network.